Fungal and bacterial infectious diseases are increasingly becoming a problem. Worldwide, 1/3 of all premature deaths are due to infections. In hospitals, multi-drug resistant infections, which are no longer affected by antibiotics, are proliferating. New drugs are urgently needed. Natural Products Research at the Hans-Knöll-Institut wants to make an important contribution. Tiny microorganisms such as bacteria and fungi harbour great potential for the development of new drugs. The unique Jena microorganisms collection at HKI contains many thousands of bacterial and fungal strains. They produce natural substances that may also be effective against infections. However, it is not easy to come by these substances. Modern methods are used for this purpose at HKI. One of them is a microfluidic system, which was developed at the Institute's biopilot plant. It allows scientists to cultivate microbial cells in tiny droplets and then examine them. Since the drops are only fractions of a millimeter large, this happens very quickly. 60,000 cultures are tested per hour. Thus, bacteria that produce previously unknown substances can be identified. These microorganisms are then examined more closely in the lab. For example, a class of so-called anaerobic bacteria that live in the soil without oxygen. A team from the Department of Biomolecular Chemistry, led by Professor Christian Hertweg, is the first in the world to succeed in inducing these anaerobic bacteria to produce antibacterial substances. By imitating their natural environment and using genetic methods, they stimulate so-called sleeping genes and can bring about natural substance synthesis. Initial tests showed that the substance is active against multi-drug resistant pathogens and is suitable for the development of new antibiotics. Junior research groups at HKI are also looking at specific communities of microorganisms for previously unknown natural substances that can be the basis for new drugs. To analyze the structures of candidate drugs in detail, they are using, amongst other things, so-called nuclear magnetic resonance spectroscopy. Using nuclear magnetic resonance spectroscopy, we are investigating the behavior of atomic nuclei in magnetic fields. This method is essentially based on the principle that certain atomic nuclei can absorb electromagnetic radiation. The information we gain from this is extremely valuable because it allows us to collect information about the chemical environment of specific nuclei. And in this way, we can explain the chemical structure of certain molecules of active substances. Equally promising for drug production are bacteria that cause soft rot in cultivated fungi like mushrooms. Hertweg and his colleagues wanted to find out what substance causes the disease and whether it is also effective against fungi that cause serious infections in humans. Mushrooms were inoculated with the bacteria and analyzed with imaging mass spectrometry. In this way, the researchers were able to find the desired substance. Such potentially active substances are then tested by the entire institute. The Department of Molecular and Applied Microbiology, led by Professor Axel Brachhager, was able to demonstrate that the bacterial agent not only causes rot in cultivated fungi, but also fought fungi dangerous to humans, such as Candida albicans or Aspergillus fumigatus.
The discovery of the active ingredient will help bring us to a better understanding of fungal disease. On the other hand, the substance is not only toxic to mushrooms, but shows a strong antifungal activity against various human pathogenic fungi. And that means that the active ingredient is also a potential candidate for the development of a drug to treat fungal infections.
To show that the antifungal drug can be produced not only in small quantities in the laboratory, it has already been made in the biopilot plant of the Institute on a larger scale. The biopilot plant forms a bridge between basic research at the Institute, that is to say, the research departments, and later on, industry. So that means we are the ones who cultivate microorganisms in the beginning, provide substances for analysis to the research departments, and who also design the bioprocesses for industrially relevant research projects. In order for the properties of the identified substances to be improved, the scientists in the Department of Biomolecular Chemistry changed the structure of substances using chemical and genetic methods. By working closely with industry partners, they can quickly translate their results into applications. Thus, life-threatening infections and multi-drug resistant pathogens could already be better addressed in the near future.